First, I would like to thank the organizer for inviting me to talk to you about what we're doing in terms of live gut microbiome and cryopreservation and assays. And I'm Daniel Figeys. I'm the CEO and director of the Quadrum Institute. Just give you a bit of background, the Quadrum Institute works in food, microbiome, and health. And we have three strategic areas. One is healthier lives. Second one is safe food, and the third one is future food. Future food is to address challenges in terms of climate change and population growth. Across all of those, we have to be able to study the gut microbiome. And one of the challenges related to studying the gut microbiome has been the lack of assays. And a few years ago, my lab started to develop some microbiome assays to be able to do what would be done, like, for example, in drug development, where you would have preclinical assays in place instead of having to do studies in animal model initially. So what we put in place is the ability to collect live microbiome, and that's done at home. So we've developed a kit where we give individual a kit where they can use a kit at home to collect a stool sample. And that gets basically couriered to us. And when it gets couriered to us, we prep the microbiome, the stool, we extract the microbiome, and we prep it for cryopreservation. And just to give you an idea, we've been doing cryopreservation from sample from North America, and we're going to start doing some from the UK and Europe. And we've collected samples in Canada, in the US, and we've collected over 400 samples so far that are cryopreserved. And if you're interested, we're not just doing human, we've done also dogs, we've done pigs, and so on, farm animals, because there's an interest to develop assays for the microbiome of farm animals as well. Now, having the microbiome cryopreserved and being cryopreserved properly, what do we do with that? So we have a collection of live microbiome. In our case, we're working on human gut microbiome. And what we've developed is a 96 well plate microbiome assay. And what you see on this slide, I'm not sure if you can see the cursor, but every single well can be a different microbiome to which you add compounds. And those compounds can be drugs, they can be prebiotics, probiotics. We've done phage, you could do probiotic as well, and other compounds. And then we read out the effects of the compound on the microbiome, and the assay is done over 24 hours. So very similar to how the assays will be done in drug discovery, for example. And the readouts that we use are metagenomics, metaproteomics, metabolomics, sometimes ELISA, or simply measuring changes in pH or redox. And we use this to do compound microbiome interaction studies and to do precision intervention trials. And if you're interested, we've published a few papers on this assay, and it's a very robust assay that can be used to study microbiome. And the microbiome, when it's put in the well, does not drift. So it remains very similar to the initial seed microbiome. And we've cryopreserved microbiome right now for over 7 years. And when we pull out the cryopreserved microbiome and we repeat the assay, it's nearly identical. I'll give you a few examples. So we did a study where we use 6 healthy adult microbiome. And this example, you can see the individual in color-coded. And in this example, I'm showing you the microbiome treated with DMSO, which is the control and the drug that is solubilized in DMSO. So the drug is triangle, the DMSO is circle. So the first drug I'm showing you is amlodipine, and it's a drug that is a calcium channel blocker. And you can see that when we treat 6 microbiome with that drug, there's very strong differences between the controlled DMSO and the treated. Second drug I can show you is the rapamycin. Again, rapamycin, what you see is much more individualized response. Some are very close to the control, some are a little more distant. And rapamycin is an inhibitor of mTOR. The third drug that I'm showing you is levetiracetam, which is, it's unclear, in fact, the mechanism of that drug, but it's used in epilepsy. And you can see that for most of the microbiome, if not all, there's not that much difference between the control and the drug treatments. So that means that we can identify changes that are very drug specific using this assay. The fourth drug that I'm showing you is canagliflozin, and canagliflozin is an inhibitor of subtype 2 sodium glucose transport proteins. There again, you can see a very strong effect. Propranolol that many people use, which is a beta blocker. Propranolol, you can see an effect on the microbiome. And finally, fluoxetine, which is selective serotonin reuptake inhibitor, you can see very strong effect. I put those there because you can really see that there are some drugs that target human proteins, such as calcium channels and SSRI. Um, uh, the receptor for the transporter for serotonin, and they have similar effect in terms of profile and what's going on in microbiome, likely indicative that there are similarity in proteins, and the proteins in the microbiome can be affected by those drugs. I'm going to zoom in a little bit just to show you in this example. So here again, I'm showing you fluoxetine and the structure above, and next to it is duloxetine, and you can see the structure. So chemically, they are very different in structure. One is a SSRI, the other one is a SNRI. You can see that both have very strong effect. And what we did is the profile look a little similar. So when we combine fluoxetine and duloxetine in one PCA analysis, what you can see is that they fall on top of each other. Basically, the square, the triangle are always together. So that points to likely targeting a similar protein within the gut microbiome, even though they have different structure. Both, for example, in human can target the serotonin transporter in the brain. So likely there are homologs in the microbiome that are being affected. So that means that compounds are not benign and on their effect on the gut microbiome, and we can see it by measuring individual responses. And some compounds have global effect across all individuals, whereas other compounds are very specific effects. Some individual respond, some don't. You can go even deeper if you're interested in mechanism study. So I'm showing you here the butyrate pathway and the effect of drugs that are SAC level 1, so drugs that are used for cardiovascular system. And if I look at the first enzyme here, which is butyryl-CoA dehydrogenase, and the drugs, you can see that some of the drugs highlighted in blue have an effect on the level of expression of that enzyme. It's being decreased. Similarly, for the acetyl dehydrogenase, you can see the two first drug decrease the level of that enzyme, and that's the enzyme responsible for acetylation. Here we have acetate-CoA transferase, the alpha and beta subunit. You can see very, very similar profile. And you can see the two first drug on the left are decreasing the level of that enzyme. Phosphate butyryl transferase, most are not changing except the one on the right. And finally, butyrate kinase, which leads to production of butyrate. On the left, you have three drugs that have an effect, and the one on the far left is having an effect basically across or most of the enzymes. So that means that those drugs are having very specific effect on the production of butyrate. And I'm showing you this example because butyrate is one of the very important metabolite produced by the gut microbiome, because it's essential for the the energy source for the colonocyte in the gut. So whether with reduction in butyrate, there's reduction in energy being available for the gut, and it will switch to a lactic acid, and therefore it will move towards inflammation. So drugs are not necessarily benign on their effect on the gut microbiome. And using the assay that I just showed you, you can go to different level of depth in terms of understanding what is going on, and you can go at that level across individuals. And through our collections, you can go from very young individuals, we have some down to 5 years old, all the way to elderly. Finally, I'm just going to show you a few examples, a couple of examples of what we do. So the same in terms of clinical, the same assay can be used for precision clinical intervention. And there what we do is we recruit individual. We then take their microbiome, isolated the way I showed you earlier, and we screen their microbiome, in this instance, against prebiotics, against a panel of prebiotics. And we select the prebiotics that gives the best response in terms of the specific metabolite that we want to see change. For example, butyrate, tryptophan, and so on, depending on which study we're doing. And then we give it to the individual over a period of months, 3 months, 6 months. And then we monitor the change and the change before and after. And I'm going to show you a few data from one of the study. And here I'm showing you the fecal short-chain fatty acid before and after intervention. And in our assay, we were predicting that acetate was going to increase. And when we treat participant 1, it's not significant, but nearly increased. The prebiotics doesn't change. We're predicting that butyrate would increase, drastic increase in butyrate in participant 1. Participant 2 is a control, no change. Propionate predicted increase, and so on. And finally, tryptophan and indole. Tryptophan, we were predicting that it would decrease. It did decrease in the participant 1, and in the control did not. And indole increased, and indole is a metabolite of tryptophan. It went up. And whereas in the placebo, there was a slight decrease in the indole. So therefore, we can predict the changes that are going to happen in the microbiome when we do an intervention using the Rapidame assay. So it allows us to do precision microbiome intervention. Finally, based on what we're doing here at the Quadram Institute, we're going to start doing live microbiome for preclinical and clinical studies, and it's called the cryobiome. And this is going to be an international effort in terms of collecting and cryopreservation of live microbiome and ensuring standardization of assay across different sites and training capacity. This way, this will allow us to do larger scale microbiome studies, keeping the microbiomes where they were collected in the country or region of origin. And the only thing we'll have to do is ship the compound that we're interested to screen, and that will be done against standardized assay. So it makes it feasible to do microbiome assay across population, something that is very difficult to do right now. And that's it. I would like to thank you for your attention.